We are gonna start asking our panel from questions from science. Then we will pass on to law and market, and it's just really. I'll start. I have prepared a couple of questions, so I'll start actually on a structured discussion, and then in between I'll leave space to the audience to actually start to chip in to ask also questions. We have heard very much about verification and validation of clinical and laboratory assays. And in research, this is extremely important. We know how much reproducibility is actually often an issue. So when we go into the clinics, this reproducibility has the importance that actually will affect and will impact patient lives. So I'm going to ask you as a first question this reproducibility and the standards that we set in a laboratory assay. How can we actually translate it into clinics? So clinics to have standards. And so NGS, we are always talking today on NGS-based assays and protocols. And is there any international effort on this? For example, I'll give a short example. We know that laboratory assays, for example, cytokine concentration, so very standardized lab tests that are run regularly in the clinics in Europe and in the US, they have kind of a variability based on the test that you use. I learned from one hematologist here at the University Hospital Basel. So how is there any international effort, or what can we do in order to actually standardize protocols and assays, NGS-based protocols and assays, in order to be actually translated into the clinic and not only into the research or a research lab or based on a special request from a physician? Please feel free to unmute yourselves, and I'll leave it free to you to start addressing the question. Then I might steer a little bit the discussion.
I'm happy to jump in to at least answer one part of your question, and thank you for such a thoughtful question that addresses so many areas that we deal with. I think one really great example of some standardization that has come about over the years is establishing the BCR-ABL fusion detection international standard, or the %IS scale that we have. I'm not sure if anyone else on the call, I would assume Dr. Davi is familiar with it.
Yeah, no, I fully agree with you. I was going to have the same answer.
Yeah, this is, what originally happened with detecting BCR-ABL levels was that it would, we would be, having a reference standard that was established in my lab and a different one in Dr. Davi's lab. And then they would give our patients different results. For some point in time, we had a conversion parameter that we could go back and translate yourself to the established international standard, which is a universally agreed upon amount in a given sample. Now, at least in the United States, you can purchase an FDA-approved assay to detect the BCR-ABL rearrangement, and that comes already aligned to the international standard. So by aligning to what is agreed upon by all labs using this, we can hopefully give our patients the same results no matter where they get tested. Dr. Davi, do you have anything to add to that?
Well, I fully agree. There's something which is important also is to have, and we have that for PCR-ABL, but also for other molecular tests, is national quality controls, where everybody analyze the same samples, and then you see the variation in the results. And for BCR-ABL, we've done that for a long time, and there was, because this is a quantitative assay to detect minimal residual disease, which could have an impact, the way you treat the patient was extremely important. And when we started, there was a lot of differences, as you said, between different labs, but just working together made things much better, and sharing a protocol and people paying attention and having those regular controls where if you're very different from the other, there was something wrong in their lab, you try to find out what it is and correct it. And gradually, it really improved things. And within a few years, the dispersion of results really came to nothing.
That's a
And
great
as you
point.
said, it's a collective effort.
When we share data, we are sharing, for example, immune repertoire sequences, right? So this data is not really genetic. We cannot come back to the person that has the data, and potentially the clonal sequence, right? It's information that about the disease that it would be used publicly at the end, right? So if it's like a medical test, you will need that sequence to either track minimal residual disease or other diagnostic purposes. So when you say data sharing and that is related to privacy of data, how we don't have, so we as an immunological community, maybe we don't have this data privacy problem that we cannot basically re-identify the individual. So how can we make it clear from a law perspective that the GDPR and all the anonymization, the identification of patient data, they don't apply to this repertoire receptor data?
Yeah, so I think that's a super interesting question. I've seen that one discussed a little bit, but I think there's basically three questions. The first one is, looking at the data on its own, right? And generally speaking, if you look at the characteristics of that information, I think you're right that it wouldn't be considered identifiable. Second question is, what happens if you use it in combination with metadata? What happens if you're, yeah, Felix, please dive in, or I can finish speaking.
No, keep talking. Sorry.
Yeah. Okay, brilliant. So then, the second thing you could think of is the combination of that data with metadata. So whether it's at the patient level, the sample level, whether it's at the level of the cohort, or alternatively, whether you're linking that data to other data repositories that pertain to, the same person. And then lastly, sort of I guess the third important question is, okay, so once you release this data, either in a controlled release or in a totally open release, how is that going to affect the identifiability? So as a community, really, I think the ways of, working through that, and I think it would be valuable even to produce some kind of whether it's a working paper, something else, is really to, present the scientific argument for why the data at the scientific level can't be taken and used to re-identify the individual alone or in combination with the other data. The second question is to look at the kinds of metadata or the kinds of data linkage that are typically performed, and then to form an argument as to whether there's anything that you are always going to need in making that information scientifically useful that would require it to be combined with, other data elements that might themselves increase the identifiability. In, thinking through those concepts, right? These questions of scientific identifiability are relatively not controversial. Once you get more into some of the genetic stuff, there's these issues of, okay, so, there's family level re-identification, or even inferential re-identification, where you have you can probabilistically prove that two people have similar data sets, or that this data is more likely than not to be a particular person's. So once you get into that sort of probabilistic space, it gets a little bit more messy once you're really looking at it in that way. But generally speaking, the scientific case is the easy one to make. The harder part of the argument, right, is when you're looking at basically scoping a release level, framing a release level from the totally open on through registered, make an account on a website and gain access to the data, on through totally controlled, have a very sophisticated access committee review every request. And that's where it becomes more difficult to determine how much, just how much basically re-identification risk is, crosses you into that threshold from, well, we can release this, on through actually, we shouldn't release this. And I think that's really, it's a subjective argument that you draw. And you really want to show that, within your data set, there's less than a 10% chance, of an individual being re-identified from the use of that data. And that's, of course, a sort of arbitrary subjective measure, but really it's about making an argument that it would be really too cost intensive to re-identify a person, or outright impossible, within the context of the data release. And then the last thing I'll say to that is that there's some quantitative measures that are starting to come out to really frame some of these risks. So you can think of, k-anonymization, which is it's a quantitative test that you use to say, well, each individual in this data set has identical data elements relative to the ones that we're considering potentially identifiable. So, everyone's basically got similar
same clone, yeah.
Exactly.
In a disease, for example, yeah.
Exactly. So those are some ways of framing that argument. And then beyond that, I mean, it's a judgment call, right?
That's great.
Not. Yeah.
Thank you so much, Alexander. So we have 10 minutes left. I'd like to make the most of it and also give the community a chance to ask you guys directly. I see there is a question online. I'll take that later. And Felix, do you have a question?
Well, yeah, I appreciated all the points that Alex made. Just wanted to mainly add that we had this Horizon 2020 grant for AIRR-Receptor and other projects, and we got a lot of hassle about AIRR-seq data being identifiable, in combination with this. So mainly maybe responding to me to Letty saying, well, in general, this express data isn't going to be under regulation, but our ethics board really gave us a lot of trouble about it. So anyway, it's certainly a big open question.
That's a mean European board. And for the scientific part, again, I've seen examples so far, or we've seen examples so far of leukemia, of malignancies, and so on that have CLL. So they are very related to B and T cells specifically. Why don't we have other clinical applications in infection or this is only an outlook, as Professor Davi showed? Why do you think this is missing? Because there's scientific papers covering all range of diseases, and then we see always the same kind of clinical applications or clinical physician requests for that.
I would say that at least in our laboratories in the United States, we have expanded our testing menu, as well as requests that we receive to cover molecular detection of infectious diseases, as well as germline genetics. We do everything from viral load monitoring to HPV genotyping, as well as we run the FDA-approved cystic fibrosis Illumina next generation sequencing test. So for the menu that we offer in the adult population, we've certainly tried to expand to cover other areas of disease. I will say that I trained in the pediatric setting, so I think you see a lot of those diseases clinically more in pediatrics, especially when we talk about germline genetics. And those are the folks that are going to have the requests for different things, such as exome and whole genome sequencing, to be launched clinically there.
Professor Davi, do you have any?
Yeah, I would say that the comment I would have is a lot of what we do is driven by a clinical need. So, what I do, to say assessment, the diagnostic of a lymphoproliferation, if you want to know whether it's malignant or no, it's absolutely essential. Then what it's malignant, if the repertoire analysis provide additional information, yes, it's important. And I'm certain that things that are not used now, which is the peritumoral, the microenvironment and T cells, the repertoire might be very important, and maybe in a few years that will be dependent because that will show whether such a tumor will respond or not to a drug. But we need this kind of proof, even experimental and done a research context. But if it's proven to be important, then there will be development to use it in a clinical context for all patients.
How far are we from that, a number in terms of time? And then I'll pass on to Felix and then to Susanna for the conclusions.
Well, it depends. If you're lucky and you find that such biomarker is absolutely important, it could take a few years. Otherwise, it could be longer.
Let's estimate. We have experts here. So in terms of time, based on the research we know is being performed and based on the regulation that we know is required, what do you think? How long before we see one of these research labs or companies translating what we find in research into the clinics, widely accessible. So not only for a few connoisseurs or people that know exactly what they need. So how long before it's large scale or even accessible from different physicians?
I think it's hard to reply. One thing is one has to make sure that the clinicians are aware that the technology is available and can be interesting, which is not always the case.
Would you dare giving an estimate, even if I understand the question is not exactly fair, but?
Well, there are two things. Either you want to, I think the best way to do it is use clinical prospective or retrospective protocol, and you have a biomarker. You think, yeah, I have a new technology to demonstrate and to detect this biomarker, and yes, take 500 patients, and those who have that kind of repertoire behave differently, and then it becomes very interesting. So you go from scientific to a more medical evaluation, and if it's statistically significant, then the community will be interested and say, well, we need really this test because it makes a difference.
Great, thank you for
that. And
that.
then once you've done, so it just really depends, how easy it is. It could take a couple of years. If you're lucky, yes, the biomarker will be extremely pertinent. Otherwise, it could take more time.
Longer. Okay, thank you for that question. It has been, I'd spend the whole night speaking about this, or the whole day for the guys in the US, because it's extremely important, timely, and we all need a community effort in that, because nobody can take this complexity by their own, and a larger community, which means a global community, US, Europe together, and all the rest of the world. Is there any question from the audience, a quick one, or a burning curiosity? Otherwise, I'd leave it to Susanna for the conclusions. And thank you very much for being with us here. It has been great hearing your presentations and having the opportunity to ask you some questions. And we look forward to keeping this network and connections active for the next years to actually translate this research into the clinics.
Thank you very much to the speakers and the moderator for this fascinating session. I think these are very important topics that we discussed today, and it's clear that we have a long time before we can say AIRR-seq has reached the clinics mainstream. Possibly there's already people going through certification processes and we don't even know. Maybe there's something going on there, but as Janos said, maybe this is going to take 1 year, 2 years, and maybe we will discover soon. Maybe in our next meeting in Porto, we'll learn of some new diagnostics. That would be very exciting. I hope you found the session useful and interesting. If you have burning questions or things you would like to discuss, maybe those would be also good topics for the podcast of the AIRR Community that is hosted by Ulrich Sterbo. And you can send us an email and we will be happy to see what we can do about that. Thank you also to Pam Borghardt, who has been recording and managing the session, and other members of the meetings subcommittee. And Carlita, thank you for helping us putting together everything. That's all. Thank you.
Susanna, do you have that slide on Porto? Because I always forget the dates and how beautiful Porto is, and I'm looking so much forward to next year, because
let me see if I have
it might be a really great opportunity to kick off some really nice things.
And just a point that the diagnostic working group is open to, anyone who wants to join and work with us in the AIRR community. So contact Ulrich or Susanna, I guess, huh?
Yeah, that's it. 3-6 of June. Now I got it.
Yes, save the day.
Thank you so much again, Susanna, for all the effort, and Ulrich, and Felix, as usual, a pleasure.
Thank you.
Wish you all a great day or a great evening here in Europe. Bye-bye.
And
Thank you.
Ciao.